I'll just click on the thing. Yeah,
Yeah, just I guess spacebar is fine. Yeah. Okay, well, please welcome Theo Portlock. He's a research institute from the research fellow, sorry, from the Liggins Institute. So please, go ahead and introduce your research.
Yeah, kia ora, ladies and gents. Yeah, my name is Theo Portlock. So I was born in the UK, where I did my studying at King's on sort of biochemistry, structural biology. So, I know a few of you are interested in sort of the 3D modeling and especially in the AR, VR space. So a little bit of that, but the fourth year down the track, I've sold the structure of a few proteins, and basically AlphaFold came out, and I was able to solve all the structures of the proteins that I had solved in my previous 4 years in a weekend. So I very quickly realized that I had to either jump off the ship or go down sinking with it. So I quickly moved to the sort of gut microbiome. There was a lab next door that was interested, and I thought, it sounds like a very interesting and emerging new field. And so that's where I got stuck in, and I moved to Stockholm, where I did my first postdoc in a place called Scilifelab. And from there, sort of moved towards the systems biology space and how different, immune system interacts with the gut microbiome, interacts with the oral microbiome, interacts with the brain. And from there, I started. Oh, this is the wrong presentation. Is this the CS one? Maybe it's not actually. Sorry. No, it's the correct one. It's the correct one. No, it's okay. Sorry. So yeah, started becoming a bit more interested in AI and how it can sort of understand the fundamental molecular mechanisms that support human health and disease. So that's a little bit about my background. But today I'm going to talk to you about a project that I've been working on with my boss, who's Justin O'Sullivan, who is the director of the Liggins the Road. It's funded by the Wellcome Leap, which is a subgroup of the Wellcome Trust that's based in the US. And it's really focused around this problem of malnutrition. So I'll do a brief intro of problems. Malnutrition is a global crisis. It's not something that only lower middle income countries face, although it does affect them disproportionately. It's made worse by climate change, is made worse by things like pandemics, people who have in the housing crisis, it's affected by cost of living, all of these factors, and it has really long enduring repercussions, not just to sort of weight and height, but also notably to sort of brain function, how able they are to perform in school and to achieve academic success, and the rest of it. So yeah, 45% of all kids' deaths worldwide are attributed to malnutrition. It's an enormous, scary number. Malnutrition is bad for a lot of reasons. It's very cyclic. So, you might have some malnourished kids. They will the families will struggle to afford the food, which will cause malnutrition. The malnourished kids grow up and they are less able to perform well in school, which means that their job opportunities later in life are affected, which increases poverty, which increases malnutrition. So there's this vicious cycle. I realize this is very biological intro, but I'm trying to make it a bit more as computer science here as possible, so keep you all entertained. So yeah, I mean, to be honest, lots of malnutrition worldwide is largely on the mend. You can see there's a slight bump, but you see during the COVID time, malnutrition slowly got worse. It's mostly Southern Asia, which is where the city of interest for my study took place. So actually, the top 3 highest rates of malnutrition globally are in sort of India, Pakistan, Bangladesh. So people think like Africa is the worst, but per capita, actually, it's you have a lot more malnourished kids in those 3. And actually, Bangladesh has largely that after a lot of external effort, to break these cycles, Bangladesh is now working very hard to make it so that these kids are growing up healthy and is doing better than it ever has done. But the problems still remain in that. Oh yeah, I'm going to take questions, right?
No, I'm just going to ask, is it possible for you to stay?
Of course. Yeah, let me move.
Slides and
Sorry, mate. Yeah.
probably
Yeah, a little bit. Al can still see. Yeah, I'm sure. So yeah, as I was saying, the short-term benefits of recovery of malnutrition are well and good, but the problem is the kids are still growing up unable to achieve well in school. Their brains are still not developed properly. When people say when development doesn't happen when it's supposed to happen, then it doesn't happen. So it's these kind of lasting problems that people are struggling to deal with. So the study itself is focused in a city called Dhaka, which is the capital of Bangladesh, and it's 154 malnourished kids, 1 year old, and 74 that are healthy. And we wanted to sort of look at the biggest differences between the malnourished kids and the healthy kids, or the well-nourished kids. And so we extensively phenotyped them all. We have lots of different data about like what was in their blood, their serum, their plasma lipids. We also recorded data in the brain, and we looked also at their gut microbiome. So that's not just like what species are there, but also like what are they doing down there, like the potential genetic components of those bacteria. So I'll start with the microbiome. So in the body, there are a number of sort of clocks that all develop, and they develop at different rates. And the gut microbiome clock develops sort of in a kind of predictable way. At the beginning, there aren't any species. When we come out of your mum, there's no bacteria in your store in your gut. But then you get the pioneer species moving in. You get oxygenation of the gut decreasing, so it becomes more anaerobic, and you see certain species like bifidobacterium, they drop out, and then you get your sort of the healthy bugs, the good bugs come in. Those are sort of the fecalibacterium. The names don't matter, but there's large systemic changes to the community structure of the microbiome, especially in the first couple of years, until you get to about 200 or maybe even 400 species at your adulthood, where they're all sort of happily living together in your gut. It's the largest sort of community of bacteria in your body. People say we're outnumbered 10-to-1 bacteria to human cells. That's true. But actually, what they neglect to say is there's 100,000 times as many bacterial genes as we have human genes. So we're really outnumbered in terms of their biology. Okay, so yeah, the microbiome, it's very important that it develops properly. And this is a study done by a bloke called Jeffrey Gordon in the States. And he basically looked at how is that aging of the microbiome early in life impacted by restriction in nutrition. And what they saw was, well, firstly, they built a sort of a random forest model to a regression to predict their age based on the species, the combinations of species that were present from the bacteria. And they could see, you can sort of work out this microbiome age, and they called it this microbiome age Z score, which is the difference between their microbiome age and their chronological age, like how old you are after birth. And what they found was it doesn't really matter when the kids were given sort of this refeeding, if they were trying to sort of recover from their malnutrition, their microbiomes were left sort of immature. So there was this lasting damage to the microbiome that wasn't affected by either refeed methods, so giving them extra calories or whatever. So in our cohort, we saw largely the same thing. Here, each dot is basically a kid, and the sort of separation between the dots represents the change in community structure of bacteria between the healthy and the malnourished kids. Some statistics show that they're significantly different in their composition of their bacteria, and that was driven because of basically two species. There's this Prevotella copperi and there's this Bacteroides fragilis. Again, the names aren't important, but this Bacteroides fella doesn't get on with Prevotella. They have this sort of, you can describe them in this sort of ratio, and that Prevotella to Bacteroides ratio seems to be significantly different. This is the biggest driver of the microbial differences between the two populations of kids. And on a genetic level, like what sort of genes those bacteria have, you can sort of look at the pathways of those genes and to see how they might function if those pathways were active. And you could see like sort of these pyruvate fermentation pathways, they were enriched, they were higher in the healthy kids. So the ability to convert this pyruvate into propanoate seems to be an important distinction between the two groups. So propanoate is it's called a short chain fatty acid. It's important for human development and of the immune system and of the brain. So these pathways are important. So this is the differences in the gut, part one. So part two, we looked at the brain. It's one of the kids from the studies. We have this sort of EEG setup where we basically put electrodes on the kid's head, very comfortable. It's not invasive, there's no drilling. And we measured sort of the direct changes to the power of certain frequencies in certain regions of the brain. So we looked at all these four regions, and what we basically found was in the temporal and frontal region at the front, there was a decrease in amplitude of the what's called the gamma and the beta waves. These are high frequency EEG measurements, but people think the high frequency ones are mainly to do with sort of the higher cognitive function, so things like language, communication, and things that require things that aren't sleeping. Sleeping is generally the lower ones, and they seem to be massively impacted by the malnutrition. So we also looked at sort of their behavior. You know, how are the kids acting when they were measured? So we measured them using this technique called the Bailey scale. Bailey is basically a series of tests. Sometimes, you know, for communication, there might be a communication test where they'll be shown a picture of a dog and they go woof, and that's a sign that they're okay, good. You know what to do. You know what's right. I mean, for these kids, they're only 1 year old, so it was more like just mimicry, you know, of the mother or of the clinician who was doing these measurements. And what we found was their ability to communicate, their expressive communication skills was very significantly different. And this is after adjusting for covariates like sort of breastfeeding and their sex and you know, what was the other one? There's three covariates that we measured to adjust to compensate for, and across the board, these scores were massively impacted. So their motor skills and expressive communication and their vocalization, we did another test to show basically the same thing, and we see massive p-values, well, these tiny p-values. And yeah, these sort of skills they sort of originate from these frontal regions of the brain and of those same frequencies. So we're painting a picture. Okay, so that's part two. Part three is the blood. So we looked at sort of fats or lipids that are in the blood, in the circulatory system. So we collected all the samples. We ran this. We put it in this fancy machine called an LCMS, a mass spectrometer. It looks at sort of the chemical breakdown of different molecules that are present in the sample. And what we found was, okay, it's a complicated picture, but the long and short of it was we saw these sort of odd chain fatty acids that were being sort of enriched in the healthy kids, and they were depleted from the malnourished kids. So we're like, okay, well, what does that really mean? So we have a big bag of all these differences. We also saw, you know, these different classes of molecules that were different. So these ceramides, were significantly lower in the malnourished kids. So ceramides, as the name might suggest, like the cerebellum, that part of the brain, they're important for healthy human development. Kids need this molecule for their education, for healthy neurocognitive. Okay, so summary of changes. There's a lot of changes I threw at you. There's changes to the gut bacteria. There's changes to the brain, the way that it's the function of the brain, the structure of the brain, the performance of the brain, and there's changes to the blood. So that's it. Except no, it's not. We can't end just by looking at a bag of differences. The important thing is to put those changes in context and work out what connects those differences. So this is where AI is important, and this is where I wanted to sort of show you how you can connect using sort of multi-omic analysis techniques, all these different data sets. So these kind of data sets are very different nature of data. So EEG, you have positive and negative values. For the gut bacteria, it's quite sparse data. You see a lot of zeros, which makes things very complicated. And for the blood, for the lipids, you get this crazy these crazy spikes in amplitudes, and you have to be very careful about the way that you analyze it. So these are very different natures of data sets. So combining them to try and understand how these changes are connected is something that it's a very complicated process. So we simplified the process a little bit. We just sellotaped all the data sets together, and we ran a Spearman correlation, the Swiss Army knife of statistics, to see which features were associated with one another. So we have a lot of connections between each of the data sets that were just associations. They were Spearman correlations. I'll go into them in a bit more. So we collected those. We looked at the context of the changes. How did malnutrition rank in terms of the other data sets? So this is what's called explained variance. So how well does this label explain the variance in the gut microbiome? It significantly explains it, and it turns out that malnutrition alone significantly explains the variance in most, not all, not the EEG, of the data sets. So we're looking at sort of the environmental context as well. And finally, we move to the AI. So this is where we switch to a random forest approach to predict malnutrition based on each individual data set. So we went through each data set and we predicted it, and it turned out the plasma lipids have the highest AUC rocks. They were the best predictors. And all the way at the bottom, we have like the gut species, the gut pathway. These are all the gut data sets. These are all the brain ones, and this is the blood. So, as a microbiome person, I was a bit disheartened by this. 0.54. There's nothing to write home about. 0.04 over flipping a coin is not something you're very interested in. So I was about to cry and go home, but we continued. So then we ran an ensemble model, a multimodal model, where we took the merged data sets and we pushed them through the random forest again, and we looked to see how well it performed in the prediction. We saw that we got some pretty good AUC rocks. I put this in this morning just for you. This is after the fivefold cross-validation, and these were the sort of performances. The AUC rocks pretty good, recall's not great, but it'll do. So it's not gonna. This wasn't really the predictive power wasn't really the final outcome of this, because then I moved to this SHAP value analysis. I'm sure most of you actually know what this is better than I do, but for me, SHAP analysis is a way to look under the black box, under the hood of the black box of the algorithm to really work out why an AI made the decision that it did when it was doing its prediction. And so we calculated all the SHAP values for all the features, and we actually compared them to their sort of linear differences, as in after just doing one versus the other. So the significance value there. And what we saw was a certain set of features were more important for the prediction than their linearity might suggest. So it sort of points to the idea that some non-linear combination of these features are important for the prediction of malnutrition. So we were a lot more happy about that because we saw some species finally popping up as being important, but they weren't linearly different, or their AUC or their sort of explained variance wasn't very good. So we looked at sort of the overall the SHAP mean for each data set, and actually we saw that the gut species and the gut pathways explained a lot of why the model made the decision that it did. So we're like, okay, what's going on? Why is it now that the gut bacteria are becoming more important? So we looked at another method called a SHAP interaction values. So a SHAP value is really it's a combination of its main effect plus all of its interaction effects for the prediction. So we looked at what those interactions were between the data sets, and we could see that sort of the gut species and the pathways then they were becoming important because they were interacting with these other data sets. So then we have the Spearman correlations I was talking about before, and now we have the SHAP value and SHAP interaction values, and we merge these together using network analysis and clustering to try and find out a mechanism of malnutrition throughout the body. So what we saw was sort of the species down here, the Bacteroides, part of that ratio, these seem to be important in predicting, as in the size of the node here is sort of the importance for the prediction. The color is their direction. So if it's blue, it means it's healthy. Orange, it means that it's malnourished and associated with malnutrition. And the edges are the red edges are the positive Spearman correlation. Blue edges are the negative Spearman correlation. And so we can see this mechanism throughout the body that eventually ends up with their communication skills right at the top of the brain. You can imagine this as like a human body here with the head at the top and the butt down there. So yeah, we saw that Bacteroides fragilis, with its fermentation pathways, were interacting through these serum lipids and odd chain fatty acids to influence EEG and the brain function at the top. So we have this. I can make this a bit more simple. So we made a simpler sort of network to demonstrate this more clearly. And these guys were in opposition to these MAM-associated species, which have these glycolysis pathways that sort of worked against in the opposite direction. So we're using the best of network analysis plus a model interpretation, plus the Spearman correlation to try and find out why these features interact in a way to cause the disease. And yet you should, of course, plot each of these out individually to make sure that you're not fooling yourself. There's a bit of stargazing with the Spearman correlation, but you see all this a lot with sort of microbiome data, but you can see the positive correlation with the expressive communication and with these fermentation pathways. So in summary, we saw that sort of the Bacteroides in the gut were associated with sort of the ceremoids and the odd chain fatty acids in the blood and their production. And those molecules were important for brain development in kids. So what this means is that even though largely, malnutrition is on the mend in Bangladesh, improved recovery methods to really boost neurocognitive function in the kids is the next frontier. It's very important to break this cycle of malnutrition. And this is the first step in the direction of creating new refeed methods to sort of impact gut species community composition and work towards a healthier nation. So I mean, this is actually part of another study. This is the baseline. So it's this and this samples from this refeed study. So basically, they eventually we'll have the rest of this data. It's still being collected right now, but they've been given two different refeed methods, and we're going to look to see if their brains are recovering better in one versus the other. So we get to see how that damage is being repaired, because the first step in fixing any problem is to really understand the problem, across multiple systems. So yeah, I mean, in summary, okay, I guess I'm a little bit early. It's 12:30, but there'll be plenty of room for questions. But in summary, stand on the shoulders of giants. That bloke at the start that who laid their groundwork for the microbiome age. That's something that came out of the top of his head, and it's important to be able to know what is important for your research. So always do that. Second, nothing changes in a vacuum. You know, you shouldn't just look at one system and think, okay, this explains everything. You need to look at as much data as you can, with exceptions. And then finally, we can't fix what we don't know is broken. So if you know, now we know how that malnutrition affects these systems, so we should know what it looks like when they are fixed. And I just, I want to really emphasize that this isn't just a one-man band. These projects are enormous collaborations between the Boston Children's Hospital and Bangladesh, of course, and also we're here at the Liggins at the University of Auckland. So I want to thank all of those people for their time and their contributions, and I want to thank all of you guys for your time. And I'm happy to take some questions.
Thank you for the presentation. Do we have any questions? Maybe also online, like open the chat window if we have questions.
Just a really basic question about nutrition. I mean, is it just lack of calories mainly that you're looking at? Is that right? Or
yeah, so that's a great question. There's a few ways to measure it. So the way that they measured it, where they classified a kid as malnourished or not malnourished is with this sort of measure of malnutrition. It wasn't just weight; they didn't just put them on a scale. It was this ratio called the length to height Z-score ratio, where basically how tall were they for how heavy they were, and if they were below 2 standard deviations, sorry, below 1 standard deviation below the mean of the average kid, then they were classified as malnourished. But I mean, most of them were underweight as well, but yeah, that's how malnutrition was sort of classified. But yeah, they weren't getting enough calories, and actually malnutrition it's not just about not having enough calories. Sometimes there might be protein malnutrition where they don't get enough of a certain like they call it kwashiorkor, or a certain nutrient where they'll have different sort of symptoms. Maybe they'll have like swollen stomachs versus a host of other issues. So it's a great question, and it's important to nail down.
I didn't quite. So with your networks, can we go back to that? Big network there.
Yeah.
So can you? So you kind of the nodes are the measurements,
Yeah.
right? And you draw a line when there's sort of a significant interaction based on the SHAP value, and
just the correlation.
Just the correlation
is the line.
Right. So what does the network give you apart from like a visualization?
So a couple of things. Firstly, I mean, you can do well here. We use Leiden clustering, which sort of looks at clusters of nodes that interact with one another by their edges, and the other thing is about the degrees and the centrality. So like how many other edges that are connected to it. So how central is it in the network? How important is it? So for the refeed method, for instance, if a node was super central in this fatty acid biosynthesis cluster of functions, then maybe that would be the one that you would target more for a particular refeed method. But yeah, absolutely, visualization is one of the main.
So what are the most central nodes there? I mean, to give some ranking of.
Yeah, I didn't show them on here, but some of these glycolysis pathways were super important, and you can see they were like very negatively correlated. So these ones would be the ones that were to target, and they come from these Rothia, Streptococcus, and Prevotella. So those species, if you were looking at a targeted antibiotic to sort of break those central nodes, for those species, then it would disrupt the whole system. Yeah, there's another network here which I didn't show either, which is the SHAP interaction network, where instead of the edges being the correlation, they are how those features combine to influence the prediction. So if you see this plus this, you're more likely to see it predicts as MAM. This is just one alone, right. And those you see particularly between Rossi and Streptococcus, when they're both present, you see a much stronger prediction of malnutrition. So it looks kind of like it's it needs two to tango when it comes to, even though individually they're important, their combination effect is more than either alone.
It's a question, but does that mean that some of these bacteria that are developed in malnourished kids are actually detrimental to the development of some certain behaviors?
Yeah, that's a really good question. So I think people tend to think of bacteria or the microbiome as like the largest sensory organ that we don't understand yet, where it just is sensitive to different sort of features. But what you're asking is how important are those? Do they actually cause an effect? That you can only really tell by either you take a mouse, germ-free mouse, so a mouse that has all its bacteria in its gut removed by antibiotic, and you give it a cocktail of those species. And if you saw that they lost weight, then you would say, okay, those species are actually actively doing something that's causative, that decreases their weight. But it's kind of difficult to get that causal analysis from a single time point. It's a good question. But in fact, there are studies for a kind of malnutrition, anorexia, in adults, where they'll take anorexic stool microbiomes and they feed them, because mice feed on stool. That's part of what they do. There's a name for it, but they graze on dropping basically. And when they did that to anorexic mice, they started losing weight. So there is some evidence behind it. The first studies, the early studies of the gut microbiome is the super early ones were for obese mice, where you'll give obese mice microbiome to another mouse and they got fat. So there's some evidence there that it influences those kind of measurements. Good question.
I have a question about the machine learning model of choice. So it seems like from the beginning you decided to use random forest, and then afterwards you said ensemble learning, which I think basically you merge different models of random forest.
So when I say ensemble, I don't mean ensemble model. I should have been more precise with you guys. No. So I mean, from the top, we use I actually used PyCarray to work out what was the best model of choice. It was extra trees random forest, and it wasn't an ensemble model as such. It was just a multimodal model, as in multiple data sets to predict the same thing.
I just want to know if you tried other models or like deep learning, or is it down the pipeline, or maybe from literature you found that random forest performed the best for this data set?
I mean, generally random forest does pretty best for microbiome data for a number of reasons. One is because a lot of the features are correlated. You see a lot, random forest is very insensitive to features that correlate. You don't have to strip it right down. And the other thing is that you can have a lot of features with random forest and it's still pretty well. So right now, this is only just over 200 samples, but we have somewhere in the region of 2,000 features. So that's not a good ratio, but it's still able to just compensate for these enormous sort of feature to sample ratios. So a few reasons. Did you have a question before? Or?
I mean, similar questions here. I always wanted to ask the machine learning models.
Okay. The question on everyone's lips.
You sort of said that it's very high dimensional, like the number of data points are under.
Super low,
Have you tried anything like Lasso?
Lasso was part of, so in the PyCarray thing, the cheat code of machine learning, it used the Lasso algorithm as well as a few others, and it didn't perform as well. We wanted the tree-based one as well because SHAP is a SHAP tree explorer that explainer that it's very high performance.
Thank you for online.
Did you have another question?
Oh, I was very curious about the refeed.
Refeed, yeah.
When they do the refeed, what are they actually doing, and what are intervals of development?
Yeah, so that's a great question. So for the remaining part of the study, or for the refeed study, it is basically they get a sachet of chickpeas, and for one of the arms, and in the other arm they have this enhanced refeed method where it's going to be company secret, but they have it twice as frequently, so they'll have 2 a day. Versus 1 a day, and it's basically to start off, they just try and get the calories up. So they just give them high calorie food, and then as soon as they reach that healthy category of -1 standard deviation of malnutrition, they get swapped to these micronutrients. So things that have like vitamins and stuff that they have been missing throughout the rest of their diet. So, but the problem is, of course, they share them with their siblings. And the families can sometimes get in the way, and you can't administer it. So refeeding is a little bit complicated, and it's also painful. It's not a nice process to the kids as well. Coming out of a disease like it's incredibly uncomfortable, and I think even it's very low, recovery rates. Only like 50% recovery of their weight after refeeding. So it's really it's a nasty problem. I've seen anyone.
You have any more questions for Theo? Otherwise, we'll be careful a bit. Okay, yeah, so thank you, Theo, for sharing your research with us. Yeah, so I'll upload this on YouTube.
That's great.
If you are good with that.
Yeah, of course, absolutely.
Right.
Thank you. It wasn't too biology.
No, it was interesting. I think
Yeah, applications.
yeah.
It's applied AI.
Yeah.
That's the